Cells under the microscope. All right. Today we have access to many powerful technologies for deciphering the principles that govern the structure and activity of the cell. But cell biology started without these modern tools. Think about the earliest cell biologists began by simply looking at tissues and cells and later breaking them open or slicing them up, attempting to view their contents. What they saw was to them profoundly baffling, a collection of tiny objects whose relationship to the properties of living matter seemed an impenetrable mystery, the same way that probably protons, electrons, and neutrons seem very abstract relative to the way we think about classical physics. Nevertheless, this type of visual investigation was the first step towards understanding tissues and cells, and it remains essential today in the study of cell biology. Cells were not made visible until the 17th century when the microscope was invented. For hundreds of years afterwards, all that was known about cells was discovered using this instrument. Light microscopes use visible light to illuminate specimens, and they allowed biologists to see for the first time the intricate structure that underpins all living things. Although these instruments now incorporate many sophisticated improvements, the properties of light, specifically its wavelength, limit the fineness of detail these microscopes reveal. Does anybody know what that's called? Like the optical diffraction limit or something like that, right? There's a limit that you just can't bend the light any more than you can after a certain point. I forget what it's called exactly. Yeah, I think it's just called the diffraction limit or something like that. Optical diffraction limit. I think microscopy is one of the coolest things to study, but it's very complex, and it's all about optics, which is physical science, not chemistry or biology. But the two definitely, this is where they meet in many ways. Electron microscopes, invented in the 1930s, go beyond this limit by using beams of electrons instead of beams of light as the source of illumination. Because electrons have a much shorter wavelength, that makes sense, these instruments greatly extend our ability to see the fine details of cells and even render some of the larger molecules visible individually. In this section, we describe various forms of light and electron microscopy. These vital tools in this modern cell biology laboratory continue to improve, revealing new and sometimes surprising details about how cells are built and how they operate. The invention of the light microscope led to the discovery of cells. By the 17th century, glass lenses were powerful enough to permit the detection of structures invisible to the naked eye. Using an experiment or an instrument equipped with such a lens, Robert Hooke examined a piece of cork, and in 1665, reported to the Royal Society of London that the cork was composed of a mass of minute chambers. He called these chambers cells, based on their resemblance to the simple rooms occupied by monks in a monastery. The name stuck, even though the structures Hooke described were actually the cell walls that remained after the plant cells living inside them had died. Later, Hooke and his Dutch contemporary Antonie van Leeuwenhoek were able to observe living cells, seeing for the first time a world teeming with modal microscopic organisms. Can you imagine being the first person to ever see cells microscopically? What a joy that would be, that you were the first person that was able to actually see a living cell. It's like one of the defining moments in human history. What a lucky group there. For almost 200 years, such instruments, the first light microscopes, remained exotic devices, available only to a few wealthy individuals. It was not until the 19th century that microscopes began to be widely used to look at cells. The emergence of cell biology as this distinct science was a gradual process to which many individuals contributed. But its official birth is generally said to have been signaled by two publications, one by the botanist Matthias Schleiden in 1838, and the other by the zoologist Theodor Schwann in 1839. Schwann was actually a very important person. In these papers, Schleiden and Schwann documented the results of a systematic investigation of plant and animal tissues with the light microscope, showing that cells were the universal building blocks of all living tissues. So this is not too far in the distant past. This is 200 years ago, maybe somebody's great-grandfather. Not that far ago. Less than 200 years ago. Their work and that of other 19th century microscopists slowly led to the realization that all living cells are formed by the growth and division of existing cells, a principle sometimes referred to as the cell theory. The implication that living organisms do not arise spontaneously, but can be generated only from existing organisms was hotly contested, but it was finally confirmed. We'll talk about that in a second. Figure 1.5, we were talking about new cells formed by growth and division of existing cells. In 1880, Edward Strasburger drew a living plant cell, a hair cell from Tradescantia flower, which he observed dividing in two over a period of 2.5 hours. Inside the cell, the DNA, which is black, can be seen condensing into chromosomes, which are then segregated into the daughter cells. This is amazing that this is from 1880, about 140 years ago. A comparable living plant cell photographed through a modern microscope. We're going to look at that in a second and see how they differed. Almost the same, right? This is an actual photograph, and this is at a 50 micrometer resolution, so well below the limit of the naked eye. All right, so this cell theory is finally confirmed in the 1860s by an elegant set of experiments performed by Louis Pasteur, and Pasteur is really, really important in the history of biology. The principle that cells are generated only from pre-existing cells and inherit their characteristics from them underlies all of biology and gives the subject a unique flavor. In biology, questions about the present are inescapably linked to conditions in the past. To understand why present-day cells and organisms behave as they do, we need to understand their history all the way back to the misty origins of the first cells on Earth. Darwin provided the key insight that makes this history comprehensible. His theory of evolution, published in 1859, so we're getting a little closer to modern times, explains how random variation and natural selection give rise to diversity among organisms that share a common ancestry. When combined with the cell theory, the theory of evolution leads us to view all of life, from its beginnings to the present day, as one vast family tree of individual cells. Although this book is primarily about how cells work today, we'll encounter the theme of evolution again and again. Light microscopes reveal some of the cell's compartments. If a very thin slice is cut from suitable plant or animal tissue and viewed under a light microscope, it is immediately apparent that the tissue is divided into thousands of small cells. In some cases, the cells are closely packed. In others, they are separated from one another by an extracellular matrix, or ECM. This is important. This is a dense material often made of protein fibers embedded in a gel of long sugar chains. ECM. I want you to remember the extracellular matrix. It's kind of what is in between cells. Each cell is typically 5 to 20 micrometers in diameter. 5 to 20 micrometers in diameter. We talked about that earlier, but I want you to always remember the size of these things and the weight. And we're going to talk about weight at some point. But these numbers matter a lot. You're not going to see a lot of numbers in biology, but I think that it's important when you do encounter numbers that you remember them. It's very embarrassing when you ask somebody how big a cell is and they're not sure. So understanding the scope of some of these things, like how big is a methane molecule and how big is a cell, it's important. The same way you could tell me how big a baseball is relative to how big a baseball stadium is. I want you to be able to do the same thing with things that are really small. All right, if care has been taken to keep the specimen alive, particles will be seen moving around inside its individual cells. On occasion, a cell may even be slowly changing shape and dividing into two. Distinguishing the internal structure of a cell is difficult, not only because the parts are small, but also because they are transparent. This is the most important concept here in microscopy. And mostly colorless, right? One way around the problem is to stain the cells with dyes that color particular components differently. Alternatively, one can exploit the fact that the cell components differ slightly from one another in refractive index, just as glass differs in refractive index from water, causing light rays to be reflected as they pass from one medium into another. Let's take a break to read question 1.3. You've embarked on an ambitious research project to create life in a test tube. You boil up a rich mixture of yeast extract and amino acids in a flask, along with a sprinkling of inorganic salts known to be essential for life. You seal the flask and allow it to cool. After several months, the liquid is as clear as ever, and there are no signs of life. A friend suggests that excluding the air was a mistake, since most life we know requires oxygen. You repeat the experiment, but this time you leave the flask open to the atmosphere. To your great delight, the liquid becomes cloudy after a few days, and under a microscope you see beautiful small cells that are clearly growing and dividing. Does this experiment prove that you managed to generate a novel life form? How might you design your experiment to allow air into the flask, yet eliminate the possibility that contamination by airborne microorganisms is the explanation for the results? There's a classic experiment by Louis Pasteur that shows the answer. But of course, this experimenter would have made the error that allowing that oxygen in would also allow microorganisms in, so they did not create life. They would have to pump in pure oxygen from a vacuum or something like that. Let's look at figure 1.6. Cells form tissues in plants and animals. This is panel A. Cells in the root tip of a fern. The DNA-containing nuclei are stained red, and each cell is surrounded by a thin cell wall, light blue. The red nuclei of densely packed cells are seen at the bottom corners of the preparation. This is cell panel B. Cells in the crypts of the small intestines. Yes, they're called crypts. Isn't that crazy? Crypt cells. Each crypt appears in the cross-section as a ring of closely packed cells with nuclei stained blue. The ring is surrounded by extracellular matrix, the ECM I mentioned earlier, which contains the scattered cells that produce most of the matrix components. All right, so light rays deflecting from one to another. The small differences in refractive index can be made visible by specialized optical techniques, and the resulting images can be enhanced further by electronic processing. As shown in figures 1.6b and 1.7a, typical animal cells visualized in these ways have a distinct anatomy. They have a sharply defined boundary, indicating the presence of an enclosing membrane called the plasma membrane, sometimes also called the cell membrane, but plasma membrane is probably the better phrase. The large round structure called the nucleus, this is also a term we use in physics and chemistry, not to be confused though. The nucleus is prominent near the middle of the cell. Around the nucleus and filling the cell's interior is the cytoplasm. These are all really important words. I haven't bolded a lot of stuff in this book, but the stuff I'm bolding now is really critical. The cytoplasm is a transparent substance crammed with what seems at first to be a jumble of miscellaneous objects. With a good light microscope, one can begin to distinguish and classify some of the components in the cytoplasm, but structures smaller than about 0.2 micrometer, about half the wavelength of visible light, cannot normally be resolved. Points closer than this are not distinguishable and appear as a single blur. So that's kind of the diffraction limit I was talking about. If you know your wavelengths, you know that red is what, around 400 nanometers, which is around, or exactly, 0.4 micrometers, right? And then going all the way to blue or violet, it's around 800 nanometers. So when people say what's your favorite color, I say around 600. But the point is that by using light, that's the best we can do with the wavelength to use diffraction. So we have to use something less than 400 nanometers. And eventually, when you get small enough, you can use something really, really small. In recent years, however, new types of light microscope called fluorescence microscopes have been developed that use sophisticated methods of illumination and electronic image processing to see fluorescently labeled cell components in much finer detail. The most recent super-resolution fluorescent microscopes, for example, can push the limits of resolution down even further to about 20 nanometers. That's the size of a single ribosome, which is a protein, a large macromolecular complex in which RNAs are translated into proteins. So the ribosome translates RNA into proteins. Interesting. That sounds really important. Ribosome. So 20 nanometers is not quite the size of an atom, right? But it's the size of sort of a protein. So we think about like micrometer seems to be cell scale. Nanometer seems to be protein scale. And maybe this is sort of the high nanometers, and low nanometers / picometers is the atom scale. And we don't really go below the atom scale, right? That's the smallest scale that we care about. Then maybe the millimeters is the organ scale. Organs are made up of cells, so that's one way to look at it as well. Organs and tissues. So these are sort of different scales that we can think about in biology. All right, let's look at these techniques in 1.7. Some of the internal structures of a cell can be seen with a light microscope. A cell taken from human skin and grown in culture was photographed through a light microscope using interference contrast optics. This is A. We see a nucleus, we have the plasma membrane, and the cytoplasm. This is a 40 micrometer scale. So the entire width is maybe, 200 micrometers. This is a pigment cell from a frog, stained with fluorescent dyes and viewed with a confocal fluorescent microscope. The nucleus is in purple, the pigment granules are in red, and the microtubules, a class of protein filaments in the cytoplasm, is green. All right, the fine structure of a cell is revealed by electron microscopy. For the highest magnification and best resolution, one must turn to an electron microscope, which can reveal details down to a few nanometers. I have one in the back. Preparing cell samples for the electron microscope is a painstaking process. Even for light microscopy, a tissue often has to be fixed, that is, preserved by pickling in a reactive chemical solution, supported by embedding in a solid wax or resin, cut or sectioned into thin slices and stained before its view. The tissues in figure 1.6 were prepared in this way. For electron microscopy, similar procedures are required, but the sections have to be much thinner, and there is no possibility of looking at living cells. When thin sections are cut, stained with electron-dense heavy metals, and placed in an electron microscope, much of the jumble of cell components becomes sharply resolved into distinct organelles, separate recognizable substructures with specialized functions that are often hazily defined with a conventional light microscope. A delicate membrane, only about 5 nanometers thick, is visible enclosing the cell, and similar membranes form the boundary of many organelles inside. The plasma membrane separates the interior of the cell from its external environment, while internal membranes surround most organelles. All these membranes are only 2 molecules thick. 2 molecules thick. With an electron microscope, even individual large molecules can be seen. So we're going to look at the fine structure of a cell can be seen in a transmission electron microscope, or a TEM, T-E-M. A, the thin section of a liver cell, showing the enormous amount of detail that is visible. Some of the components to be discussed later in the chapter are labeled. They are identifiable by their size, location, and shape. So this is a peroxisome, this is a lysosome, this is a mitochondria, and this is the endoplasmic reticulum, or the ER. There's the plasma membrane, and there's the nucleus. Let's see what's in cell B. A small region of the cytoplasm at a higher magnification. The small structures that are clearly visible are the ribosomes, each of which is made of 80 or so individual protein and RNA molecules. Some of the ribosomes are free in the cytoplasm, while others are bound to a membrane-enclosed organelle, the ER, endoplasmic reticulum, discussed later. I'd say ER is a really important concept. So this is in panel B. We have the ribosomes, the endoplasmic reticulum, and the mitochondrium. Mitochondria, plural. This is at 2 micrometers, so this is really zoomed in. This is 2 micrometers, but not as zoomed in. So here's 50 nanometers, so this is extreme zoom, and we can see a DNA molecule. And again, it's very zoomed out. It looks like it could be the surface of Mars or something, right? Portion of a long thread-like DNA molecule isolated from a cell and viewed by electron microscopy. So that's what panel C was. The type of electron microscope used to look at thin sections of tissues is known as a transmission electron microscope, a TEM. This instrument is, in principle, similar to a light microscope, except that it transmits a beam of electrons rather than a beam of light through a sample. Another type of electron microscope is a scanning electron microscope, SEM, which scatters electrons off the surface of a sample and so is used to look at the surface details of cells and other structures. These techniques, along with different types of light microscopy, are reviewed in panel 1.1. Even the most powerful electron microscopes, however, cannot visualize the individual atoms that make up biological molecules. To study the cell's key components in atomic detail, biologists have developed even more sophisticated tools. Techniques such as X-ray crystallography or cryo-electron microscopy, or cryo-EM, important idea, relatively new, for example, can be used to determine the precise positioning of atoms within three-dimensional structure of proteins and their complexes, which we'll discuss in chapter 4. How big are cells and their components? This chart lists sizes of cells and their component parts, the units in which they're measured, and the instruments needed to visualize them. These drawings convey a sense of scale between living cells and atoms. Each panel shows an image that is magnified by a factor of 10 compared to its predecessor, producing an imaginary progression from a thumb to skin cells to a mitochondrion to a ribosome to ultimately a cluster of atoms that forming part of one of the many protein molecules in our bodies. Note the ribosomes are present inside mitochondria as well as in the cytoplasm. Details of molecular structure shown in the last two bottom panels are beyond the power of the electron microscope. So let's start with our thumb. This is somebody's thumb, and we take this little piece and magnify it to from 20 millimeters. This is very visible, right? 20 millimeters, all the way to 2 millimeters, which is less visible, but we can see some detail there. And then finally to 0.2 millimeters, which is 200 micrometers. And now we're looking at, we can see individual cells, which we couldn't see before. Now at 20 micrometers, we can not only see individual cells, we can see some cell components. At 2 micrometers, we can actually see an entire cell component. And we can see the mitochondria, for example. We see ribosomes, but not in good detail. At 0.2 micrometers, or 200 nanometers, we can now see the ribosomes. At 20 nanometers, very, very small indeed, we can see the actual ribosomes and even little parts of the protein. So these are proteins. And at 2 nanometers, we see the proteins. At 0.2 nanometers, these are individual atoms. This is the angstrom length. So we can see the scale here, and I would highly recommend you study this and think about it closely.